Hello friends, and welcome to CEC online lectures. Friends, we know that there are certain chemicals in our body which are synthesized in our body, and they control various physiological processes as well as growth and development of our body, and we call them as hormones. Likewise, in plants also, such kind of chemicals exist or are being synthesized inside the plant body, and they control their physiological processes as well as growth and development, and we call them as plant hormones. So friends, this is what I am going to discuss in my new lecture series on plant hormones. Now in this lecture series, we are going to discuss whether the plant hormone is the correct term or we should use plant growth regulator. Then what are these plant growth regulators or plant hormones? Then we will discuss different types of these chemicals with special emphasis on auxins, gibberellins, cytokinins, abscisic acid, ethylene, brasinosteroids, polyamines and jasmonates. And we will discuss what are various effects or the physiological effects that are exhibited by these chemicals or these growth regulators or plant hormones on the plant body. So friends, coming on to the correct nomenclature or the correct term for these chemicals. Now if we talk about the term hormone with respect to animal physiology, then it is a chemical messenger which is synthesized at a local site in the body. Then it is transported to the specific site of action with the help of systemic circulation, and at that particular site of action it exhibited its effect, which leads to or which includes the control of different physiological processes including growth and development, and this effect is largely concentration dependent. So as I told you that similar kind of chemicals are also found in plant bodies, wherein they are synthesized at a localized site or they are transported through the vascular system of the plant to the site of action, and then they exhibit their effect which control various physiological processes in the plant body, and therefore the term plant hormone was given to these chemicals when these were discovered. But in 1981, Trevor gave certain contradictory statements, or he highlighted certain contradictory properties which were the major reason for giving the term hormone, because the term plant hormone was given on the basis of the character of these chemicals inside the animal body. So he gave certain contradictory properties of these chemicals. He stated that plant hormones, though they have a localized site of synthesis, but their synthesis is widespread. It occurs in various cells and tissues. There are certain hormones which are transported to their site of action, like cytokinins. They are transported from roots to the leaves where they prevent senescence. But there are certain hormones which exhibit their effect within the cells where they are synthesized. Say for example, ethylene is there. It exhibits its effect in the cells where it is synthesized. Then it was stated that animal hormones, the effect of the animal hormones is concentration dependent, whereas Trevor stated or he exhibited that the effect of plant hormones is not purely dependent upon the concentration, but it is also dependent upon the sensitivity of the tissue towards that particular hormone. So some of these contradictory statements or contradictory observations made by Trevor made the scientific community to think whether the plant hormone is the correct term that should be used for these kinds of chemicals, or we should give some new term, and therefore various other terms were given like plant growth substances, but this term was not widely accepted because of the vague term substance that was used in this terminology. Then a new term was given, plant growth and development substances. Again, the vague term substance was used in this term, and moreover the plant growth is only one aspect of the development of the plant, because these are the substances which can also exhibit inhibitory effect on the plant growth. So therefore this term was also not widely accepted. Then came the term plant growth regulators, and this was the most widely accepted term for such kind of chemicals, because regulators is a broad term which also covers the inhibitory effect as well as the developmental effect of these chemicals, and therefore plant growth regulator became the most widely accepted term for such chemicals. Another term, phytohormones, was also given, but this was not widely accepted. As of today, plant hormone as well as plant growth regulator, these are the two terms which are most widely used, widely accepted terms, and these terms are interchangeably used. So in this lecture series, I'll be using the term plant hormones. So what are these plant hormones, or what are these chemicals that are termed as plant hormones? Now these are the group of naturally occurring organic substances which influence physiological processes. As I told you, that they control physiological processes of the plant body at a very low concentration. That there are three main phenomena. These are not the only phenomena, but these are the three major phenomena that are being affected by these hormones, and these include growth, differentiation of cells in different tissues, and development or the morphogenesis, that is the development of different types or different organs of the plant. So these are the three major physiological phenomena that are being affected by hormones. Obviously there are certain other phenomena like opening and closing of the stomata which are also controlled by these hormones. Another elaborated definition of plant hormone was given, and according to this definition, these are the organic substances other than plant nutrients, active at very low concentration, and are formed in certain parts of the plant and then translocated to other sites where they evoke specific biochemical, physiological, and or morphological response. So this is a more elaborated definition of plant hormones which covers almost every aspect of these chemicals. Now we move on to the first class of the plant hormones, that is auxins. Now auxins are the first plant hormones that were discovered, and the term auxin has been derived from a Greek word auxein, which means to grow. Now in 1880, Julius von Schausch, who is considered as father of plant physiology, he gave a very interesting observation, and his observation laid the foundation for the research on these plant hormones. According to his observation, he stated that there are certain chemicals or substances in the plant body which are responsible for the growth of the plant, which are responsible for the development of the plant parts. And this observation, as I told you, formed the basis for the research in this particular field. In the same year, Charles Darwin, who was also interested in the movements of the plant, specifically phototropism. So in the same year, he published a book, The Power of Movement in Plants, wherein he explained certain observations of his experiment. He was working on coleoptile of a canary grass, and he found whenever he exposed the plant towards the source of light, he found that the growth of the plant was towards the source of light. So in his experiment, he covered the tip of the upper part of the canary grass with a foil, and then again exposed that particular plant towards the light. And he found that at this time there was no curvature or no turning of the plant towards the source of light. Then he covered the lower part of the plant and he again exposed the plant towards the light, and at this time he again found that the growth of the plant moves toward, or the plant moves towards start growing towards the source of light. And he hypothesized that there are certain substances that are present in the upper part or the tip of the grass which makes them to move towards the source of light. Now, following his lead, a Dutch botany student, Fritz W. Wendt, he started working following the lead of Charles Darwin, and in 1926 he started working on oat plant, that is Avena sativa. So in his experiment, what he did was he cut the tip of the coleoptile of Avena and placed that tip on an agar block. Now after a specified period of time, he again removed that agar block and placed that agar block on the stump of Avena, from where the coleoptile has been cut off, or the apical part was cut off. And he found that again the growth was restored as if a new tip has been placed on the stump of the Avena. And he hypothesized again, and he established that there are certain growth substances that are present in the apical tip of the Avena. And his findings eventually lead to a very famous test, which is known as Wendt's Avena coleoptile curvature test, or Avena test, in which he again removed the tip of the coleoptile and he placed the agar block and found there was a curvature in the plant and the growth was restored. And the curvature of that particular plant was directly proportional to the concentration of the auxins that were present in the tip. Later, Kogi and Hagensmith in 1931 isolated the first auxin, though this isolation was not from the plant but from the human urine, and they gave the term auxin, they gave the name to that first isolated auxin as auxin A. Later, Kogi, Hagensmith and Exerben, they again analyzed the human urine, and then they isolated another type of auxin, that is auxin B. Though auxin 1 was first isolated, so they again isolated auxin 1 and auxin A, and in addition to auxin A, they also isolated auxin B and along with an heteroauxin. Later, the heteroauxin was identified as Indole 3 acetic acid, which is the major auxin or the naturally occurring auxin. Then Kogi and Kosterman in 1934, they isolated Indole 3 acetic acid from yeast. Then Thiman in 1935 isolated Indole 3 acetic acid from the culture of fungus Rhizophus suinus. And in 1946, Hagensmith, the same group, they reported the auxin that was isolated from the higher plant. So this was the first report in 1946. The first report came wherein the auxin was isolated from a higher plant. And now we know various natural auxins that exist, and the most common examples are Indole 3 acetic acid. As I told you, this is the most widely occurring natural auxin, followed by 4-chloro-indole 3 acetic acid, phenyl acetic acid, Indole 3 butyric acid, Indole 3 propionic acid. Now, Indole 3 butyric acid and Indole 3 propionic acid are considered to be very less active or almost inactive auxins. Likewise, synthetic auxins, the most common examples are 2,4-dichlorophenoxyacetic acid and naphthalene acetic acid. These are the two most active synthetic auxins. And another example, 2,4-dichlorophenoxybutyric acid. It is a very less active or almost inactive synthetic auxin. Now we move on to the biosynthesis of auxins in the plants. Now, auxins are biosynthesized by two major pathways, which are named as tryptophan dependent and tryptophan independent pathways. And amongst these two, tryptophan dependent pathway is considered as the major pathway for the biosynthesis of auxin, and it is the most widely studied pathway. In tryptophan dependent pathway, the synthesis of Indole 3 acetic acid can occur through 4 intermediates, and these 4 intermediates are Indole 3 acetyldoxine, Indole 3 acetamide, Indole 3 pyruvic acid and tryptamine. And amongst these 4 intermediates, the pathway that followed the intermediate Indole 3 pyruvic acid is considered to be the major pathway of the auxin biosynthesis in the plant. So as you can see that tryptophan, after being synthesized by the indole, it gets converted into tryptamine by tryptophan decarboxylase enzyme, which causes decarboxylation of tryptophan, thereby converting into tryptamine. Now tryptamine is acted upon by UCA enzyme, which are monoflavin type of enzymes, flavin monooxygenase type of enzyme, and get converted into N-hydroxytryptamine, which finally gets converted in indole 3-acetatoxine. Now tryptophan, by action of CYP class of enzyme, can directly get converted into indole acetatoxine. Now this indole 3-acetatoxine gets converted into indole 3-acetic acid either by indole 3-acetaldehyde as an intermediate or indole 3-acetonitrile as intermediate. But as I told you that the major pathway is through indole 3-pyruvic acid, so tryptophan also gets converted into indole 3-pyruvic acid by TAA enzyme, or also known as tryptophan transaminase enzyme, which causes the deamination of tryptophan. Now this indole 3-pyruvic acid undergoes decarboxylation into indole 3-acetaldehyde, which is acted upon by UCA enzyme or indole acetaldehyde oxidase enzyme, and finally gets converted into indole 3-acetic acid. So this is basically how the indole 3-acetic acid, or the major auxin, is biosynthesized in the plant. Now we move on to the transportation of the auxin. Though it is synthesized, now how it is transported to the site of action. Now if we look into the overall transportation of auxins, it is considered as polar transportation, that is from the apical region to the root tip, from the aerial tip to the root tip, which is underground. And if we look its movement in the shoot system, or in the part of the plant above the ground level, then the movement is basipetal, that is from the apical or the tip of the shoot to the base of the shoot. And if we look into the movement of auxin in the root area, which is below the ground, then such movement is acropetal, that is from the base of the root to the tip of the root. But overall the movement is polar, that is from the tip of the shoot to the tip of the root. Now we move on to that how this auxin is transported within the cells. Now if we look into the structure of the plant cell, the outermost region is the cell wall, followed by the cell membrane or the plasma membrane, and then the cystole, where all the organelles are there. Now the cell wall matrix is having an acidic pH of somewhat around pH 5 or 5.5, and this is largely due to the high concentration of proton ions present in the cell wall matrix, whereas cystole has a basic pH of 7. Now indole 3-acetic acid occurs in two forms in the plant cells, either in the protonated form, where it is known as IAAH, or the ionized form, which is IAA negative. Now the protonated form, that is IAAH, can easily diffuse through the plasma membrane and enter into the cystole, whereas the ionized form requires specific carrier proteins or the influx carrier proteins, which help them moving inside the cystole. And these carrier proteins are nothing but Auxin or lax proteins. I will not go into the detail of subtypes of lax proteins, but one can remember is there are two types of protein, Auxin and the lax protein, which are the influx carrier proteins and allow the ionized form of indole 3-acetic acid to enter into cystole. Now this protonated or ionized form of indole 3-acetic acid has another property, that under acidic condition this ionized form immediately gets protonated, because as I told you that cell wall matrix has higher amount of protons, therefore the ionized form easily get protonated, and this protonated form, that is IAAH form, easily diffuses inside the cystole. And once it enters into the cystole, as I told you that cystole has a basic pH of around 7, this IAAH get ionized or deprotonated. So it releases the proton ion. Now these proton ions are pumped out of the cystole region with the help of proton pump, which utilizes ATP as a source of energy. And these proton pumps then again enter into the cellular matrix and thereby maintaining the acidic pH of the cell wall matrix. Now this ionized form of indole 3 acetic acid cannot cross the plasma membrane. Therefore, they require another class of protein which are known as efflux carrier protein, and these are known as pin 1, pin 2 or pin 3 proteins, which will help or which will allow these ionized form of indole 3 acetic acid to cross the plasma membrane and again enter into the next cell. And once they cross the plasma membrane, they enter into the cell wall matrix where the pH is acidic. Again, they get protonated and they easily diffuse into the next cell. So this is how the transportation of auxins takes place in a vertical direction from the apical tip to the root tip. And this largely occurs through the phloem parenchyma. Now we move on to the physiological actions of auxins. Now auxins, the major action that is exhibited by auxins is the cell elongation or increase in the length of the plant, because as you elongate as you increase the length of the cell, the overall length of the plant also increases. Now what happens is, as I told you, that auxins cause an increase in the excretion of protons with the help of proton pumps, which utilize ATP molecule as a source of energy. Now this acidic pH of the cell wall matrix causes activation of xyloglucan endotransglycolase enzyme. Now this enzyme help in hydrolyzing the bonds of xyloglucan, which is a hemicellulose compound. And we know that plant cell walls are largely composed of cellulose, hemicellulose, pectic substances and so on and so forth. So why I am emphasizing on xyloglucan is because this is the major component which is responsible for holding the cell wall together in addition to cellulose and other compounds. So once these xyloglucan bonds are broken, the cell wall gets loosened up. And in addition to this, the acidic pH that is due to the increased proton concentration in the cell wall matrix because of action of auxin also activates the expression of certain proteins which are known as expansin proteins. And these expansin proteins also help in loosening of the cell wall. Now, auxin not only stimulate the proton pumps to pump out the protons into the cell wall matrix, but they also stimulate the potassium channels, which causes the influx of potassium ions into the systole in order to balance the alteration in the charge that has occurred due to the excess proton effluxing. Now once the concentration of potassium ion increases inside the systole, it also causes the influx of the water inside the systole. And whenever the water start moving inside the systole region, it causes increase in turgor pressure, it causes expansion of the cells because walls have already been loosened up, so the cell basically expands, and this expansion is irreversible, and eventually this expansion leads to the increase in the size of the plant cell. So this is how basically the auxins cause an increase in the size or size of the plant cell, or causes the elongation of the cells. Next action of the auxins is the differentiation of vascular tissues. Now it has been observed that auxin stimulates the differentiation of certain cells, vascular cells, into the respective tissues. And it has also been observed that whenever auxins are applied in a higher concentration, they causes differentiation of the xylem cells into xylem parenchyma, xylem vessels, xylem tracheids and xylem fibers, which are the four components of the xylem tissue. Likewise, if auxins are given in low concentration, then they causes differentiation of phloem tissues into phloem parenchyma, into sieve tubes, companion cells and phloem fibers. Now this differentiation of vascular tissues occurs through two major pathways or the phenomena. One is reaction diffusion pre-pattern, and another theory is on the canalized Auxin flow. And amongst these two, canalized Auxin flow is the major theory which is widely accepted that explains the differentiation of the vascular tissues. Then auxins are also used as herbicide because they also inhibit the growth of certain plant tissues. At normal concentration, as I told you, it has been found that auxins cause increase in protein synthesis, increase in DNA and RNA content synthesis. But at higher doses, this increased protein synthesis or DNA or RNA synthesis goes beyond the acceptable levels, and it eventually disturbs the normal DNA to RNA ratio and normal RNA to protein ratio, because it has been found that the Auxin treatment increases the rRNA concentrations in the tissue. So this disturbed DNA RNA ratio or RNA to protein ratio eventually disturbs the cell division, which finally prevents the growth of the plant. And therefore it is normally auxins at very high concentration or at specified concentration, they are used to kill the weeds that are growing in near vicinity of the desired plant, and therefore they are called as or used as herbicides. Now auxins also regulate the apical dominance. Now apical dominance is a phenomena wherein the apical bud suppresses the growth of the lateral buds or the auxiliary buds. Now as we discussed that majority of the auxins, they are located in the apical bud region or the shoot apical meristem region, that is the SAM region. Now auxin, since they are present in large amount in that region, they stimulate another type of hormone which are known as strigolactone hormones or the branching hormones. And these hormones, they stimulate the expression of a protein, a class of protein which are known as branching protein, and specifically BRC1 protein or the branching 1 protein. And this protein, it inhibits the development or the growth of the lateral buds. So this is how the auxins regulate the apical dominance. And if we cut the apical bud, and when we cut the apical bud, we prevent the expression of strigolactone hormones, we prevent the expression of BRC1 protein, and hence we can stimulate the growth of lateral buds. So we can remove the apical dominance by cutting the apical bud. So this is another physiological action of auxin, that is they induce the apical dominance, and if you want to remove the apical dominance, we can cut the apical bud, which will allow the further flourishment or the growth of the lateral buds. Now apart from these major actions, auxins have also exhibited the inhibition of the primary root formation. Now primary roots are those that develop directly from the radicle. But on the other side, auxins stimulate the development of adventitious and lateral roots. So I repeat, auxins inhibit the growth of the primary root, or it inhibits the primary root formation, but auxins stimulate the development of adventitious roots as well as lateral roots. Now auxins also have exhibited the development of fruits, specifically in seedless fruits, and this phenomena is known as parthenocarpy. So we can say auxins also lead to parthenocarpy. And auxins in various tissue culture studies have shown to increase the production of various secondary metabolites. Now apart from these physiological actions exhibited by auxins also interact with other growth hormones or the plant hormones. And auxins have shown in respect to cytokinins that their levels are inversely proportional, and auxin treatment can rapidly inhibit the cytokinin biosynthesis. Whenever we are exposing a plant to an exogenous auxin, it increases the expression of the gene which is responsible for encoding the rate limiting enzyme in ethylene production. As I told you that auxin also stimulates the ethylene production, which further prevents the growth of the plants, and therefore it is also used as herbicides. So auxin, basically they increase the expression of that gene which encodes, or in simple words if I say, auxins stimulate the action of the enzymes that produced ethylene, thereby it start producing the or it increases the production of ethylene. As with the ethylene, auxins also increase the production of gibberellins, which is another type of growth hormone, and the basipital movement of auxin is necessary for the production of these gibberellins. So friends, this was about auxins, how they are biosynthesized, how they are transported in the plant from their site of synthesis to the site of action, and what are the various physiological actions that are exhibited by auxins. So friends, in the next lecture I'll be discussing about another class of plant hormone, that is gibberellins. Thank you.